The embryonic development between day 15 and 21, essentially week 3, will start our journey with the bilaminar germ disc, which is comprised of the epiblast cell layer and the hypoblast cell layer. These are two epithelial cell layers on top of each other. I like to equate the bilaminar germ disc as two layers of pizza doughs that are sitting on top of each other. At day 15, there are two indentations where the epiblast and hypoblast are pinched in together or adheres to each other. Close to the connecting stalk, we have the cloacal membrane. On the opposite end, we have the oropharyngeal membrane. Between day 14 and 15, we start to develop this raised longitudinal ridge within the epiblast cell layer called the primitive streak. This occurs by rapid cell division of epiblast cells. The rounded cranial end of the primitive streak is called the primitive node. Along the midline of the primitive streak, the cells start to ingress, causing a midline depression called the primitive groove. This is where the epiblast cells get crowded together and are pushed down towards the hypoblast cell layer. The circular ingression of the cells at the primitive node creates a central depression called the primitive pit. The ingressing cells of the primitive streak will undergo EMT, epithelial to mesenchymal transition, and they will quickly migrate in between the epiblast cell layer and the hypoblast cell layer towards the cranial end of the embryo, establishing the mesodermal layer. I like to think of this process as a magical soft cheese layer that's forming from the top layer of the pizza dough, and that cheese kind of spreading in between the top and the bottom layer of the pizza doughs, creating some crazy ravioli. This mesoderm, or cheese of the ravioli if you will, tend to migrate in 3 paired streaks. The most medial pair, or closer to the midline of the embryo, is called the Paraxial Mesoderm, and the pair lateral to the Paraxial Mesoderm are called the Intermediate Mesoderm, and the pair that's spreading most peripherally is called the Lateral Plate Mesoderm. These 3 different pairs of mesoderm will give rise to different structures in our body. Now, from the primitive node, there's no EMT occurring in the cells that are ingressing. Instead, these ingressing cells will hold onto each other and form a nice rigid cord of cells that form the nodal cord and grow towards the oropharyngeal membrane. While the nodal cord is lengthening, the mesodermal tissue also spreads towards the cranial end as well. And with the formation of the nodal cord, there's induction of the overlying ectodermal cells to thicken and form a raised plate within the ectodermal layer called the neural plate. The neural plate at this point is made up of the neuroectoderm that is specialized from the regular ectoderm. With continued tissue production from the primitive streak and the primitive node, the embryo proper lengthens and grows. So there's lengthening of the nodal cord and elaboration of that neural plate, all due to the new tissues that are being generated from the primitive streak. So the paraxial mesoderms all continue to migrate in. From the caudal end, the intermediate mesoderm grows as well. While the paraxial and intermediate mesoderm stop at the level of the neural plate, the lateral plate mesoderm goes all the way around, even cranial, to the oropharyngeal membrane and form a nice little arc at the edge of the developing embryo. And at this point, the embryo is now made up of 3 germ cell layers as opposed to just 2 cell layers. It is now referred to as the Tri-Laminar germ disc. As the Tri-Laminar germ disc grows, there seems to be some interesting things happening with the neural plate, with some folds and grooves that are appearing. So let's make a cross-section right through here and take a look at what is happening. In cross-section, we have the regular ectoderm and then the thickened neuroectoderm forming the neural plate. And the neural plate seems to have risen up dorsally on either side, forming what we call the neural folds, while the midline appears to have kind of sunken ventrally, forming a midline groove called the neural groove. Just under the neural plate, we have the nodal cord cross-section that has of course induced the neuroectoderm to differentiate from the regular ectoderm. Underneath that, or ventral to that, we have the endoderm, and immediately on either side of the nodal cord, we have the paired streak of mesoderm, the paraxial mesoderm. Lateral to that, we have a pair of intermediate mesoderm. On the peripheral most zone of the tri-laminar germ disc, we have the lateral plate mesoderm. So in 3D, we have from the ectodermal surface, neural folds that have risen up, and then the relative midline groove, neural groove that has formed, just caudal to the oropharyngeal membrane. So this is about middle of week 3, day 18 or 19 or so. Towards the end of week 3, so day 21, the embryo has grown quite significantly and its shape has also morphed quite a bit. So let's make sense of this. Right now it kind of looks like a tadpole here, but the oropharyngeal membrane is still intact and the cloacal membrane is still present towards the caudal end. The primitive streak is still present. Note that this looks small, but the primitive streak has not shrunken. It's the same size. It's the rest of the embryo that has elongated and grown cranial to this little tissue factory that could. So we have now a significantly longer notochord in the mesodermal layer, and just above that we have the neuroectoderm that has been induced to form from the ectoderm. This middle part that looks pinched, there's a reason for that. We talked about the neural folds forming, and in this mid-plane of the embryo, the right and left sides of the neural folds will come together in the midline to fuse. Let's take a cross-section and take a closer look. Here we have regular ectoderm that has risen up with the neural fold, and the neural plate that has sunken under towards the ventral side, and the tips of the neural folds that are coming together. It's still continuous with the regular ectoderm. So this neural plate made up of the neuroectoderm ends up forming this closing circular structure that bring the right and left regular ectodermal edges together, which will eventually fuse with each other, and the neuroectoderm gets pinched off. While that happens though, at the very tip of the neural folds, there's specialized cells indicated by this green color, where the cells undergo yet again EMT, epithelial to mesenchymal transition, and become migratory, while the majority of the neuroectoderm will sink under and become the neural tube, which will eventually give rise to our central nervous system, essentially the brain and the spinal cord. This new population of mesenchymal cells, called the neural crests, will form all throughout the length of the embryo, and they will give rise to so many different and important structures in our body. For this reason, the neural crest cells are considered to be the fourth germ layer in our body. They give rise to the PNS, peripheral nervous system. They form the adrenal medulla. They get into epidermis all over our skin and give rise to the melanocytes, and our beautiful facial structures are for the most part contributed by the neural crests, which differentiate into the facial bones. All the other embryonic structures are still present. We have the notochord, ventral to the neural tube, paraxial mesoderm on either side of the neural tube, lateral to that, the intermediate mesoderms, and even lateral to that, we have the lateral plate mesoderm, all dorsal to the endodermal layer. So this process of neurulation will continue to progress in cranial and caudal directions. So we're zipping the neural tube cranially and caudally. So from the dorsal aspect, this is what we'll be seeing. The regular ectoderm now covering most of the middle part of the embryo, and the neural tube that's sunken under, pinched off of the skin in the middle part of the embryo, but the neural tissue in the cranial and caudal end would be still continuous with the skin. Another important concept here is that as the neurulation is progressing, this rapidly expanding neural tissue causes the entire embryo to start to curve or fold cranially and caudally, and the lateral folding will also ensue as well. So that's where we are in terms of neurulation by the end of week 3. Now let's see what other stuff is happening in the vicinity. Here's the pair of paraxial mesoderm. They're coming from this primitive streak right next to the developing neural tube and the nodal cord. They've migrated all the way to the cranial end. Paraxial mesodermal tissues don't just stay mesenchymal. They start to segment into nice ball of tissues at a regular interval, starting from about the mid-region of the embryo, while the cranial and caudal portions still kind of remain mesenchymal. So these balled up segments of paraxial mesoderm are called the somites. The somites give rise to many important structures, including the axial skeletons in our body, the vertebral column, pelvic girdles, and the ribs, or skeletal muscles, and the dermis of the skin. The pair of intermediate mesoderm from the primitive streak migrate cranially on the lateral aspects of the paraxial mesoderms. And this pair of streak of tissues will give rise to the urogenital ridge in embryonic anatomy. Based on the name, can you guess what they might give rise to in our body? Among other things, they'll give rise to the kidneys and gonads. The lateral plate mesoderm is the peripheral most group of tissues that originate from the primitive streak. By the end of week 3, this mesoderm layer has actually split into 2 layers. The dorsal layer in contact with the ectoderm is called the parietal layer or the somatic layer of LPM, and the ventral layer in contact with the endoderm is called the visceral or splenic LPM. Visceral or splenic both refer to the viscera or the organs. And this new space that's formed between these 2 layers of LPM is called the Intra-Embryonic Cavity. Another word for cavity in embryonic anatomy is the coelom. So in this diagram up here, the LPM is spread in between the ectoderm and the endoderm at the edge of the Tri-Laminar germ disc. And unlike the other mesodermal tissues, the LPM will go all the way around the rim of the germ disc, even cranial to the oropharyngeal membrane. And within this LPM, we now have a newly formed intra-embryonic body cavity that follows the contour of the LPM. So it kind of looks like a horseshoe-shaped tunnel that runs through the LPM. So looking at what these structures give rise to, the parietal layer of the LPM will give rise to the lateral and anterior body wall. It also gives rise to the appendicular skeletons of our body, so the bones of the limbs. And the visceral LPM gives rise to a lot of important visceral structures, including the connective tissues of the visceral organs. The cartilages of the trachea, for example, gives rise to the smooth muscles. Even the heart comes from the visceral LPM, as well as the majority of the blood vessels within our body. This horseshoe-shaped intra-embryonic cavity too will give rise to the adult structures. So the pleural cavity, the pericardial cavity, and the peritoneum are all deriving from the intra-embryonic coelom.